Here are some presidential candidates telling their stand on glycolysis.
First and foremost, glycolysis occurs in the cytosol. Glucose enters the cell and is phosphorylated by the enzyme hexokinase, which transfers a phosphate group from ATP to the
sugar in the cell because of the impermeability of the plasma membrane to ions. Glucose 6-phosphate is rearranged to convert it to its isomer, fructose 6-phosphate, using ATP.
Enzyme transfers a phosphate group from ATP to the sugar. So far, the ATP shows a decade of 2. Now the sugar is ready to be split in half. An enzyme cleaves the sugar molecule into
2 different 3-carbon sugars, glyceraldehyde phosphate and a hydroxyoctane phosphate. The next enzyme in glycolysis uses only glyceraldehyde phosphates as its substrate and
is unperceptive to the other. An enzyme now catalyzes 2 sequential reactions while it holds glyceraldehyde in its active site. First, the sugar is oxidized by the transfer of
electrons and H+ to NAD+, forming NAD. Finally, glycolysis produces some ATP. The phosphate group added in the previous step is transferred to ADP in an exergonic reaction.
For each glucose molecule that began glycolysis, step 7 produces 2 molecules of ATP, since every product after the sugar splitting step 4 is doubled. By the end of step 5,
glucose has been converted to 2 molecules of 3-phosphoglycerate. Next, an enzyme relocates the remaining phosphate group. This prepares the substrate for the next location.
An enzyme forms a double bond in the substrate by extracting a water molecule to form phosphonylpyruvate or PEP, making it unstable. Now move on to part 2.